Okay, so I'm Vanessa Pilati from the University of Oviedo in the Universidade de Brasília. I will present my work as a postdoctoral researcher at the University of Oviedo, which is called Superparamagnetic Mn-Ferrite Nanolabels for radiofrequency inductive detection in magnetic lateral flow immunoassays. We all know how helpful rapid diagnostic tests based on lateral flow assays are. They are very useful, and we have been using these tests since the 80s for detecting pregnancy, for example. And during the pandemic, they were very useful for detecting the infections of SARS-CoV-2 virus. But we can also find these tests for detecting bacterial infections, cancer biomarkers, and also another diseases. The lateral flow assays are based on microfluidics and biorecognition. We have a paper device. We deposit some drops of a fluid to analyze in the sample and conjugate pad. In the conjugate pad, we have some biorecognition molecules that can capture the analytes together with a nanolabel or a nanobead or microbead to be seen. And this system will flow by the pores of the nitrocellulose membrane until finds the test line. In the test line, we have another biorecognition molecules that can capture the analytes together with the nanoparticles. And the rest of the fluid goes on until the control line where we have another biorecognition molecules to ensure that the test worked properly. Another possibility is using a vertical assay where we introduce the strip, and here in this case we mix the biorecognition molecules together with the fluid to analyze, and the system will work the same way. The previous configuration is most used in the commercial lateral flow assays that we can buy, for example, in a pharmacy, but this configuration of the dipstick assay have some advantage, and this is why we are using in our studies. The results we all know, if we have two lines, if we have the analyte in the sample, we have two lines, if not, just one line. Most of the commercial lateral flow assays are based on colloidal gold nanoparticles and microparticles. They provide a visual signal, usually a red line. We can see in the test line or in the control line. And sometimes we want to know the concentration of the analyte in the sample, which is the biomarker we want to detect. And it's very challenging because this is a optical based signal, and with some variations in ambient light and modifications in the color of the paper. This is very difficult to achieve. So we have just a semi-quantification of the biomarker in this kind of lateral flow assays lack of sensitivity. However, we can use the magnetism in our favor. Using magnetic nanoparticles, they will provide not only a visual signal, but also a magnetic one that can be detected and quantified using magnetic sensor. And also we can use these magnetic nanoparticles to pre-concentrate the sample, which is very interesting when we are dealing with complex samples such as the blood, where we have a lot of biomarkers. We can, using magnetism, we can separate them from the other biomarkers and also avoid inspecificity in the test. So to detect the magnetic nanoparticles in the test line, we are using radiofrequency inductive sensors in our lab. This is the sensor we have now. Here we have a planar coil and a micropositioner. This was specifically developed for reading magnetic lateral flow assays. The circuit is linked to an impedance analyzer that monitors the impedance of the circuit. This is a typical measurement of a scanning of the lateral flow assay of the test line. We can see that when the nanoparticles approach the sensing part, the sensing coil, their switching magnetic moments will produce some eddy currents at the surface of the coil, that this will increase the impedance of the circuit. So we can see here a peak of the impedance, And what we assume as the magnetic signal of the test line is the area above this peak to account for all the nanoparticles in the test line. And this is proportional to several things from the sensor, but here I am focusing on the nanoparticle, the nanotag in the test line. So here is proportional to the mass of nanoparticles. Of course, if we have more nanoparticles in the test line, we will achieve a large signal. But also this depends on the magnetic susceptibility of the nanotags. We can improve the signal for inductive detection by increasing the mass of magnetic material. This can be achieved, for example, using magnetic clusters, but we can also increase the initial magnetic susceptibility by optimizing these nanoparticles. And in my work, we are focusing on this part. So since we have an AC magnetic field, we need to account for the two contributions of the real and imaginary components of the magnetic susceptibility. This is modified with the frequency accordingly to the Nel relaxation theory in the case of superparamagnetic nanoparticles and in the absence of magnetic interactions. Here we introduce the log normal size distribution that we usually have in our samples. And here, a calculation for the range of frequencies that we operate in our sensor, which is in the megahertz, some megahertz of frequency. Here, a calculation for the case of 10 nanometers magnetite nanoparticles. We can see how this susceptibility, this is the moduli, how can be modified. If we reduce, we oxidate the nanoparticles, the magnetite to obtain maghemite nanoparticles, therefore reducing their magnetic anisotropy, we will achieve a high value of the modulus of the susceptibility. But if we reduce even more by changing the chemical composition of these nanotags, we can increase even more the susceptibility, and therefore we can increase the sensitivity for inductive detection. Another advantage of using manganese ferrite nanoparticles is that their colloidal dispersion are black or dark brown, while the magnetite and maghemite dispersions are more orange, and the gold ones that are not magnetic, of course, they are red. So in this work, we synthesized manganese ferrite nanoparticles, and we investigated their efficiency for inductive detection. We used the co-precipitation technique and functionalized the nanoparticles with citric acid to provide carboxyl groups at the nanoparticle surface to further conjugate to antibodies and proteins. We made some characterization with X-ray powder diffraction. We saw that they are pure spinel structure, which is good. We confirmed the citric acid coating by thermogravimetric analysis. We synthesized two different samples with different diameters to investigate the influence of the size in the magnetic detection. And they present a log normal size distribution. From the magnetic characterization, we can see that at low temperature, they have a very, very small magnetic anisotropy, a very small coercitivity, which is related to the small magnetic anisotropy. We can see that at room temperature, they are all superparamagnetic, the entire size distribution. We confirm this by the ZFC-FC curves and this distribution of blocking temperatures. And also we saw that the saturation magnetization that we can achieve is very close to the book manganese ferrite. We also investigated the AC susceptibility in high frequencies because the signal that we measure in the sensor depends on their frequency that we are using to measure. So we can see that the large nanoparticles present a large magnetic susceptibility, and therefore we expect a large sensitivity in the sensor. So investigate the inductive response of the magnetic, the samples. We printed different lines, different concentrations of ferrofluid to have different mass of deposit nanoparticles, and we measure the signal of these lines to mimic a test line. We measure the magnetic signal as a function of the deposited nanoparticles mass, and we can see a linear trend. The best linear fit provide us values to the slope, which is the sensitivity for magnetic detection, which represents how much magnetic signal we can achieve with the mass of nanoparticles. So we can see that the large large manganese ferrite nanoparticles provide a large sensitivity, so they are more sensitive. They are more sensitive for detection. We here compare two different magnetite nanoparticles. They are most used in the magnetic lateral flow assays, and we can see that for the case of 10 nanometers, they were synthesized using the same chemical route. They have the same size, and we can see that we have three times the sensitivity in comparison to the magnetites. And we also compare to our previous best results using magnetite nanoclusters, and the sensitivity is twice just by using manganese ferrite nanoparticles. So we investigated their performance in lateral flow tests. We used the AVGene biotin model as a biorecognition. These are two proteins that have a very strong affinity between the two. So what we did was to attach neutrovigene proteins in the surface of the nanoparticle, and we printed a biotin test line, and we put the system to flow in the nitrocellulose membrane. If we have the neutrovigene in the surface, these nanoparticles will be captured by the biotin line, and they will provide a color and a magnetic signal that we can detect and quantify. So we did this using the same parameters, same protocol, and we saw that the nanoparticles with large diameter provided a large magnetic signal as expected. So we decided to go further with these nanoparticles because they are more sensitive for detection. We optimized the protocol, investigated the reproducibility of the bioconjugation, and we confirmed the attachment of the neutrovigene molecules by using dynamic light scattering. We investigated different concentrations of neutrovigene, and we saw that this increasing the concentration, we are increasing the magnetic signal because we are increasing the amount of magnetic nanoparticles in the test line. So the maximum signal we achieved with this small 10 nanometers manganese ferrite nanoparticles were comparable to the huge magnetite nanocluster, which is very good. So to conclude, we optimized magnetic properties for inductive detection by modifying the chemical composition of the nanotags. At 20 megahertz, which was the frequency we used for our measurement, we increased the sensitivity for magnetic detection more than twice in comparison to our best previous results with magnetite nanocluster. And now we are working with detection of real biomarkers for the detection of pneumonia infections. In this the framework of this project, Pneumosensor, if you are interested to read more about it, here is the link. And also we recently published this work with the neutrovigene in nanoscale advances. Here's the QR code for those who are interested. And I want to thank to our group and our collaborators, our funding, and thank you all for your attention.
So thank you, Vanessa, for your nice presentation. So I'd like to see if there are any questions from the audience. If there are any questions, please feel free to ask them directly.
Perhaps I have a question about the quantification of the signal. So right now you are applying it in the lab with quite bulky instrument. There will be a way to have a quantification of the signal also for commercially available tests?
Well, the commercially available tests, we don't have a magnetic test yet, as far as I know. For detection of the gold nanoparticles, for example, we can use optical methods. We can take a picture, for example, with our cell phones. but this is difficult to have a precision in this quantification, because any modifications in the ambient light, the flash you use, this can modify the signal. So the standardization in this kind of tests, it's very difficult, but they exist. They are not very, how can I say, sensitive, but they exist.
Thank you very much for your talk.
I think you should.
Okay, so I guess so now we are short in time, so we just thank you very much, Vanessa, for your presentation. So I invite you to later to be in the Gather Town.